So next topic is polymerase chain reaction, and first of all we will discuss its introduction. What is PCR polymerase chain reaction? We all know that genome of other organisms, including human beings, is very big. We talk about human genome. It is made of 3 billion base pairs. Scientists often need to isolate a very specific segment of DNA within a vast amount of genetic material. And since this segment is just one tiny piece of the genome, we need many copies to have enough for the work. So what is done? We take one copy of that particular genome and we amplify it. We make 2 copies from it, then 4 copies, then we get 8 copies, and so on. So PCR is a technique by which any piece of DNA that can be quickly amplified in vitro, in lab conditions. So we can study one copy by amplifying it. This PCR was discovered by Kerry Mullers, and he visualized it during a long car drive. What he did, he summarized it in his own words, like it is beginning with a single molecule of the genetic material, DNA. The PCR can generate 100 billion similar molecules in an afternoon, in few hours. This reaction is easy to execute, and it requires no more than a test tube, a few simple reagents, and a source of heat. So these are his words. So what does it mean? It means that by having just a test tube, few reagents, and some source of heating, we can get millions and billions of copies of the desired gene in the lab conditions. And this is the very basic technique which is applied in biochemical and molecular techniques in studies in various branches of science. And since it was very important, it was basis that he was awarded Nobel Prize in 1993 on the basis of this discovery. So in summary, PCR is a technique in which we amplify the one particular gene or the gene of interest, and we get billions of copies of that particular gene in a few hours.